Today you'll be seeing a demonstration of an aliquoting technique. Why do we want to do this? Samples are collected to quantitatively measure substances of interest, for example, cortisol. Many proteins and hormones are temperature sensitive. Repeated freezing and thawing of the sample causes deterioration of some of these substances, making measurements impossible. Aliquoting, or splitting the sample, minimizes this risk. The easiest way to aliquot a sample is to use a disposable transfer pipette. You'll need cryovials. These are polypropylene vials made for storing samples at -80 Celsius. Racks to hold your tubes, labels to identify your aliquoted samples, and your sample to be aliquoted. Note how the label is applied to the tube. Samples must be homogeneous, so a vortex mixer is important. There are usually two setting options: continuous, which tends to be a little noisy, and auto setting, where a slight downward pressure of the sample triggers the action. A benchtop trash receptacle is handy, as are Kim wipes for any spills, a 10% bleach solution for cleanup, and some paper towels. Gloves to be worn for sample handling, a cryo storage box for sample organization, and of course, a clean lab coat. The CDC does not consider saliva a biohazard when there are no visible traces of blood. It's important to check your university's policy before disposing any materials into municipal waste. It's important to organize your workspace in a way that's comfortable and ergonomic. This means that the technician should be aware of their posture, keeping all equipment and supplies in close quarters so not to overextend one's reach and to avoid static positions. When doing repetitive work, there's a strong likelihood that you'll create a strain on your muscles and joints. Take a 1 to 2 minute break every 20 minutes. Organization is very important. Arrange your tubes in the order they will be aliquoted. To make life easier, uncap all the lids of the cryovials you plan to aliquot and place them someplace clean, like on a paper towel. If you plan on doing a lot of work, uncap a group of vials and store caps and vials in separate Ziploc bags. Cryovials must always remain in a rack while aliquoting. Don't let this happen to you. Now we're ready to get started. The first thing to do is to get an idea of your sample volume. If you need to record the volume collected, now is the time to record this information. Using your non-dominant hand, pick up your first sample and vortex. Remove the cap and grab a transfer pipette. Before the tip of the pipette goes into the liquid, squeeze the bulb to dispel air and place the tip into the liquid. Slowly release pressure on the bulb to draw liquid up, always keeping the tip in the liquid. Drawing up air creates bubbles, so don't do this. Place the tip of the pipette into the first vial and squeeze the bulb gently to dispense the sample. Repeat with the second vial. If you are aliquoting more than 2 vials, continue aspirating and aliquoting as needed. Ideally, samples should be divided equally among multiple tubes. Don't be stingy with your sample. Filling vials 3/4 full is acceptable. Filling the cryovial to the brim will cause the cap to crack in the freezer and results in lost sample. When you're satisfied with the volume in each tube, cap the samples. In this demo, we're using different cap colors to differentiate aliquot 1 from aliquot 2. This could also be accomplished by writing the aliquot number 1, 2, etc. on each cap. Check the ID number of the original tube and find the corresponding aliquot labels. Affix the appropriate label to the cryovials. Cryo-storage boxes are made of moisture-resistant fiberboard that are great for sample organization and freezer storage. The 9x9 insert helps with this organization. Label each row on the vertical axis A through I and the horizontal axis with numbers 1 through 9. Position the box on the bench with numbers facing you so that space A1 is in the upper left corner. Vials will be placed in each space working left to right in each row, A1 through A9, B1 through B9, etc. Each space in the box will correspond to a sample. This creates your sample roster. When you are finished placing tubes in the box, label the outer box and place in the freezer. Now you're finished.